Dear students, in this module, I'm going to elaborate on the online resources that are available for comparing protein structures. You know that multiple types of protein structure comparisons can be done. For instance, you can compare whole proteins with each other, or you can compare the portions within these proteins with each other. These portions may include small motifs, domains, right? So because domains are representative of functions, so therefore it is a very interesting study as well. More so, once you have compared the protein structures, then you can calculate their root mean square distance and talk about their similarity. Towards the online resources for protein structure comparison, the first one is the PDB-E-Fold. So this program is available on EBI, that is the European Bioinformatics Institute, and you can access it here in the URL given on top. And next, you can simply click the Launch PDB-E-Fold button to arrive at the software itself. Here I have shown you a screenshot from the software. So as I mentioned in the comparison strategy, that you can either have a pairwise sequence alignment done first in order to identify which alpha carbons correspond to each other, or you can have a multiple sequence alignment. So this will help you to align the alpha carbons in multiple proteins instead of just two. So once you have identified which type of 3D alignment you are interested in, you can choose between pairwise or multiple. So the pairwise will simply tell you about comparing two proteins, while multiple will be about comparing multiple proteins. If you look at the left side, you can see the query box in the red dotted square, and you can select the source or the query protein by looking at its PDB entry, and here you can also provide its PDB code. So if you know which query protein you are using and its PDB code, then you can input it here. Also, for the target, you can specify the target's PDB entry here, and the software will compare the structures of these two proteins using pairwise comparisons. Moreover, you can look at the chains that are there in these two proteins, and you can select which ones do you want to compare. In the end, you can sort by their scores and you can submit your query. So this was the case where you were trying to compare a query protein with a target protein, or simply two proteins. But what if you want to compare a protein with the entire PDB database? So you want to see which other proteins are similar to the protein of your interest in the entire PDB database. So for that, the software provides you with setting the source here for the target as the whole PDB archive. So if you select the whole PDB archive, then the protein that you have specified here will be compared with thousands of different proteins that are there in the PDB, and those proteins which are similar to the protein of your choice that you have inputted on this side as query will be output. So in conclusion, the protein structures can be compared in multiple ways. You may want to compare two proteins, or you may want to compare one protein with the entire PDB database, or you may even want to compare portions of the proteins with each other. And the software that I just discussed can help you do just that. Moreover, there are several advanced techniques that are there that are available as well, and we will talk about them later.